Okay, I'm going to give you a talk that's, and first, Keith, thank you for the kind words. Actually, one of the things I'm proudest of is helping to train you. So I'm going to give you sort of 4 lessons here, a quick overview, a little bit about Delta, sort of how did it all get done, research is about people, and some personal reflections about this. I don't have to say too much about it being a formidable pathogen. This is the Johns Hopkins website from yesterday, and we have 737,000 deaths. Way undercutting of underreporting of 250 million cases and over 5 million deaths, and we have passed the 1918 pandemic. So how did my involvement in COVID-19 vaccine development happen? Well, it first started actually in December when I had my common dinner with ASF, who to me is known as Tony, which other people call Dr. Fauci. When we sort of started talking about tracking what was going on with this epidemic in Wuhan and feeling that it was really uniquely human adapted and probably had legs. He went on in January, you know the story in January. And on March 3rd, Tony called me and sort of said, hey, want to sort of enact what we had talked about with respect to the vaccine development program. And his first question was, hey, come to Washington and, second yourself to the government. And I sort of said, you're at the White House, I'm going to be in the Omni Hotel. Everybody else that I work with is in Seattle, the organization's in Seattle. So over the last few days, one of the key people in my discussions was Peter Gilbert, who's the head of the computational immunology and head of the biostatistics program of the CoVPN and one of the world's great statisticians. In mid-March, I tell Tony I'm staying in Seattle. A week later, NIAID leadership agrees that the HVTN leads this effort, not announced until a month later. And by the end of sort of March, I wrote this email and really had already outlined what ended up being our strategic program for the government's conceptual framework of how we were going to do this. So, and this occurred well before Operation Warp Speed. So let's talk about how this all happened. Well, first of all, you needed to start with a vaccine immunogen. And that had nothing to do with me or the CoVPN. That was some work, and some of these slides are Tony's and some are Barney Graham, on a 20-year effort that had been in the structure-based design of viral envelopes. That actually really started with the founding of the Vaccine Research Center, which was the HIV Vaccine Research Center 22 years ago. First with the Peter Kwong being recruited and really spending a lot of time on the structural biology of HIV envelope. But Barney Graham also came in that recruitment, was interested in respiratory syncytial virus. And it was he who really sort of started to identify that it was the pre-fusion form of the viral envelope for many of the respiratory viruses that was actually the form in which neutralizing antibodies were most potent. And if you think about that, when a virus, at least some viruses, with their landing gear, hit to the receptor and close. When we do neutralization assays, we don't distinguish between fusion and pre-fusion. And you will measure neutralization, with either. Now, the epitopes are actually quite different. And the functionality actually of essentially the potency of the epitopes, really for many viruses, including coronaviruses, are more potent in the pre-fusion state. He worked this out with RSV, showed that it was pre-fusion that did it. Actually, his first constructs with pre-fusion were not stable. Just sort of think about this, what takes more energy, to keep your arms like this or like this? takes more energy, so it's more unstable. Actually, it was not stable in the bottle. Actually did a couple clinical trials, didn't work actually, and when you go back to the bottle, the protein is like this. So they then had how to stabilize this. And they worked through it with human coronaviruses. They started with SARS, they did human coronaviruses. This is Barney working with Andrew Ward, again, a very prominent HIV investigator. And this actually is, they found that stabilizing with prolines, and this is the 2SP, these beautiful spikes. And actually, this is the spring-loaded. This is what the pre-fusion protein looks like when it actually spring-loads and goes out. It shows you the wild type is a mixture. And so what we generally get is this mixture, and we measure the neutralization that way. Now, this also had been worked out that with coronaviruses. I showed you the human coronaviruses. And also when MERS came, they made pre-fusion proteins. They actually put them into the monkey model. They actually did a phase 1 clinical trial. So you essentially de-risked the concept that the spike protein in the pre-fusion proline stabilized form would work with a coronavirus. And we knew that actually before the identification of SARS-CoV-2. And it's one of the reasons that within actually 8 days of the genome being the genome being put on the envelope, this photomicrograph actually was obtained in the VRC lab, in Barney's lab, the lower right-hand panel. 66 days actually from the identification to actually 22 days to get it to Moderna, 66 days to making the first batch of RNA for the phase 1 clinical trial. Just sort of amazing. All right. The vaccine platform is the next part of that. Now that had been, again, thought about during the pandemic preparedness period of time. And the physicians and working with the NIAID at BARDA sort of selected out the fact that we were going to use multiple platforms. Many reasons for that, I'll talk about that. In which we would have a nucleic acid platform, a viral vector platform, and a traditional recombinant protein platform. It turned out that there was a little bit of a DNA platform, but that was given by electroporation, so that fell off the radar screen. So we're talking about the RNA platforms being Moderna and biotech Pfizer, the adenovirus viral vector platforms, both being in the adenovirus field with chimp addox from the AZ here is actually from the Jenner group from Oxford, and the recombinant proteins, the Sanofi and Novavax. And the timelines for this also differed greatly, but the concept was essentially to have multiple platforms. So this is a slide that I actually made very early in April for the first sort of public talk I gave about how we would approach this, is that we needed to develop multiple vaccine platforms. There was no single platform that could be scaled to manufacture for the 7 billion people globally. This was always designed to be a global program. We needed to use known platforms because we were in an emergency situation, and manufacturing scalability was a key factor. We needed to coordinate this, and there must be an unprecedented and coordinated approach to test, manufacture the vaccine at scale, and importantly, to deliver the vaccine into people's arms throughout the world. So that was the design in the first place. We outlined this actually in a paper in Science that I wrote with John Muscola, who worked with me during this. He's the head of the VRC, was the main liaison between NIH and BARDA as a government employee, and with Tony and Francis Collins on sort of defining this approach in which we would use harmonized efficacy trials. That the WHO and other people sort of conceptually sort of thought, well, you line them all up and you wait, and then you run them all against one placebo group. It's like, hey, they're never going to be manufactured at the same time. I had learned that through 25 years of working in things in vaccines. B, we needed all of them to be winners. No one wanted to compete with each other, and it was 98% or 92%. Did we really care? The point was we still needed to do this. And speed was important. So when we first started designing the trials, we thought, well, we could probably do this with 12 or 15,000 people. we sort of made the decision, hey, let's do it with 30,000 people, compress the time. That's a large enough study to make public policy. So we essentially worked on harmonizing people. What does that harmonizing the trials mean? Common endpoints. We use common clinical trial networks. We harmonized the endpoints and how people were diagnosed and followed. In fact, the UW Virology Lab with Alex and Keith is one of the Operation Warp Speed laboratories that did the virology endpoints and the quantitations associated with the trial. The correlates of protection analysis was done by the CoVPN, which means the HVTN statisticians over at the Hutch. And we also had a common DSMB that would monitor all the trials and keep it as a playing field. And that has been really a stalwart of the data that came here. So essentially, but even before Operation Warp Speed was announced on April at the end of April, and Monsef Slaoui was named OWS chair, we had this all outlined and done between that period of time. The phase 1 study started early on really in March. And Operation Warp Speed had a wonderful tenet in which it actually, the Army brought to it, which was no meeting in which which had to have a sort of a resolution or an outcome, could last more than, the next meeting had to be within 48 hours. And so nothing could be done that was, over 48 hours. And that actually is the way the phase 1 data were done. As soon as they came in and the labs churned it out, everybody, not the investigators, the regulators, and everybody in the company, all looked at the data within 48 or 72 hours, made a decision like, hey, was it safe? This is the dose, et cetera. We then move on to phase 2. We start phase 2. You do the same thing. There's no month lags here. It is on a 48 or 72 hour point of view. And that's actually the way we ran the CoVPN from beginning to end. We gave my staff 72 hours to do things in between times, but that is the national effort. All right, so we have a platform, we have a vaccine construct. Now we have to build an investigative army to do this work. For that, again, HIV was the NASA of COVID-19. And we essentially, both the technologies as well as the infrastructure, really came out of the HIV program. So the HIV Vaccine Trials Network, the HPTN, we've been working together on the antibody-mediated prevention trials and everything we've done. We joined together. There was an influenza clinical trials network from NIAID, and we worked with the VA to build a network that, when we started, we had 60 sites by June, and we built it up to 152. It was very easy and gratifying to me to call every infectious disease investigator in the Midwest and the Southwest and the Southeast and ask them to join the effort to be part of the clinical trials program. And that we were able to sort of build this investigative army in really record time. Each one of these trials that we did had about 100 clinical trial sites. About 40 of them per trial were out of academic centers, and about 60 of them were clinical research organizations that the PPD, Ecovia, the companies brought together, and we harmonized them and brought them together into each of these trials. They started the end of July, the opening day lineup here. Moderna and Pfizer have been competing since then, or within one day of each other. AstraZeneca started in the end of August, J&J in September. Novavax, the protein, was supposed to happen in October. They had a harder time manufacturing it, and they didn't start essentially till January 1st. Sanofi stumbled. Their first set of manufacturings, they actually didn't do the concentration of the protein right. It was only half a dose, so they lost about 4 months. They had to redo it again. And they actually didn't start their clinical trials till April of 2021, and they're ongoing now. Again, the proteins are great once made and once manufactured, but the time to make GMP proteins is clearly here. It's even in 2021, it is an art form. It's not easy to make. It's not easy to make under pressure, and it has it's not the platform to use, we've learned, in an epidemic. Now, we needed to recruit 125,000 volunteers. And we had an epidemic in which the BIPOC community, the black and brown community, had the highest rates of acquisition, highest rates of death, and that was related to their higher exposure, higher density in living, higher occupational exposure, and we needed to know whether vaccines worked in that arena. This was not just a casual issue of, quote, we want to do this. You needed to, this was the population we needed to protect. So how are we going to recruit 125,000 volunteers in a period of time in which we designed these trials that every trial would enroll totally 30,000 people in an 8-week period of time? So we started a website. Jim Kublin, who works with me, and Julie Ledgerwood, who worked at the VRC, worked with Oracle in setting up a national registry called the Coronavirus Prevention Network. We got a sign-up of about 600,000 people. It's now 650,000 people who said they were interested in a clinical trial. It was HIPAA compliant, so their demographic, their living situation, their employment information allowed us to sort of collect a high-risk score versus a low-risk score with respect to sort of exposures, potential exposures. By zip code, they went to the sites, and the site then could click on it, and they would know both from race and exposure history, a group of people who lived in their catchment area, and hence they could essentially call to enroll into the trials. It's still probably the best educational website. Yesterday we just put the FAQs up on booster doses, their testimonials. It is really, you want to know about COVID and COVID vaccines, this is a great website. How do you engage black and brown communities? You do that through trust. You do that through the fact that we've been working in this arena in the HIV field for 20 years. Our community engagement people, Michelle Andrasik, Stefan Wallace, Sally Bach, who is our marketing person, and Gail Broder, our educational person, really worked on this. And we were given unlimited resources. And so I actually had a marketing program. We could make TV ads, and we used social media ads. We went to the media that the Latinx and black communities used, and we engaged the BIPOC communities. We got expert panels that were trusted persons to actually read the protocols and talk about them to their constituencies. We had already a faith-based program in the HVTN. We extended this. Called COVID in Black. Stefan did this. And we had some faith-based issues with faith-based programs with clergymen as well as national black leaders, including those who are the presidents of the black medical schools. And they did one with Tony and Francis Collins, which they had 600,000 people on the webcast. So an educational program was extensive that was marketing the trials. So basically, that's what happened. And by July, we started what we'd call I called the race here, which was on July 27th. Moderna started, and July 28th, Pfizer tried it. The trials were designed like this. As I said, an 8-week enrollment period. The trials were endpoint evaluated trials, and they turned out to be 150 endpoints. The trials are actually very similar and weren't hard for us to design because these are the trials we've been designing for HIV. Now, why 150 endpoints? Because that gives you, that's enough to give you a 50% efficacy with a 30% lower bound. So, if we wanted a vaccine and that was the cutoff, we needed at least something above 50%, that the 30% bound, i.e. that the confidence limits were such that you knew that the minimum efficacy was 30%. If you had 50%, it could be anywhere from 30 to 70%. And that's the reason for that. I'll come back to the red line, which is that the actual Moderna trial was that, we met the enrollment guidelines, but the epidemic was such that we were able to accrue many more cases by the time the endpoint came. And also just to look at this exponential curve, that when you have a cohort that's this large, you get to 50 in 4 months, but you get to 200 within 6 weeks of that. So holding the trial till its end. I don't need to tell you this, you can read this, the vaccines were great. I never thought anything would be above 70%. The day I learned that the Pfizer and the Moderna trial were in the 90% was really sort of what could you do except smile and hope all your Kaplan-Meier curves look like that. And you've seen this. I'm not going to spend any time on this. We were able to do the AD26 built on our international program. And that was, we wanted to use the sites internationally. And also we were worried that we were going to use up all the sites in the United States, so we wanted international capacity. So we put the J&J trial internationally, and that was as much luck as anything, because when we went to South Africa, there was the Beta variant. And we went to Latin America, there was the Mu and the early parts of the Lambda variant. And so we got a very early read on the fact that, the vaccines could work, albeit we took a little bit of a hit. But we learned that incredibly early, and again, illustrated the global nature of it and the advantage of working globally. All right, by the end of the year, we had, science gave us the breakthrough of the year, COVID vaccines. That was great. But implementing the discovery of the year was not necessarily that easy. We learned that there were a lot of pitfalls in getting ready with respect to rolling out a vaccination program in our country. Lots of luminary people got it, but it did take a while and it did take effort. But it did work. Now, even though we got to only 50% coverage, when you mathematically model what happened, COVID vaccines saved, even at the 50% level, 150,000 lives and 900,000 hospitalizations. And what happened is that we averted the alpha virus epidemic that occurred all the way in Europe. That vaccination program clearly did that. So coverage is really important, and all the modeling now shows that a 1% increase in coverage actually is more important than a 25% increase in efficacy. Because covering people is important. So how did all this happen from, January posting of this to November actually getting the efficacy trials out of RNA? That's 11 months. Is it dumb luck? Was it cutting corners? You can read this. The answer to this is none of the above. It happens because none of it was quick. It's science and building upon science. And for me, yes, there was some luck about it, but it was sort of, I'll take the analogy that it was just in time, like manufacturing, of 4 very critical parts that all took place 20 years of work in each part to come together that we could do this like this. It is an American science success story, and it is really, quote, only in America could you do this. And it is us who did this, and Europe didn't do it, and no one else did it. So what are those efforts? Number one is 20 years in the development of mRNA as a vaccine technology, and I'll go through that. 20 years scientific effort in the structural biology of viral envelopes and the importance of the prefusion structure. I already gave you those slides earlier in the talk. There's 15 years of pandemic preparedness with SARS, MERS, Zika, and bird flu that established the preclinical animal models. We had the non-human primate models and the phase 1 human trials. This de-risks both the vaccine platform and the constructs. So we were pretty confident that this was the way to go. We put all our money, 5 out of the 6, 4 out of the 5 EUAs that we did, all have the prefusion structure to this. The only one that didn't is the chimp ad-ox. And against variants, this is having a hard time. And I think that's the reason for it. And 20 years of the HVTN as a CRO that could organize, design, and conduct the trials. And some luck, yeah. We knew that 99% of people who got COVID cleared their infection. This was not HIV, which were 0 out of 77 million, that the human immune system doesn't know how to clear HIV. We knew that the human immune system needed to do that. We knew that if we could mimic that, it would work. And it turns out we're better than it. We're better than natural infection. And we're lucky for that. Just to think about RNA, obviously discovered in the 82, but the demonstration that naked RNA is translated was in 1990. In 10 years later was the demonstration that when you did that, it elicited all these TLRs, especially TLR3, that really made translation very incredibly inefficient. But you could have this work. The key patent that both BioNTech and Moderna have is this patent filed in 2005 by Karen Carrico and Drew Weissman, and they just got the Lasker Prize. I presume they'll get the next one next year. Is in 2005 was the generation of the RNA platform, and they showed that you could modify mRNA without triggering key inflammatory pathways, especially TLR3, overcoming the key hurdle and paving the way for current vaccines. That's the really issue of the modification of RNA. The lipid envelopes is how we protect RNA, and one of the unique things that has been discovered, over the time, is that these lipid nanoparticles, thousands of them, are entering each cell, both the muscle cell and particularly T follicular helper cells in the germinal lymph nodes. And that's really probably a major issue by why you get such a good antibody response and why you get such a good CD4 T cell response. And that's been done with multiple different pathogens. Component 2, the structural biology, I already gave you the slides on that. And you can read that. That's 20 years of work. And number 3 is the congressional appropriation of pandemic preparedness, that Ebola, Zika, bird flu, RNA. Here's just an important 2016 that is actually about the Ad26 vector that came out of Dan Baruch's lab. That its vector, we had worked with it in HIV, but had done a lot of work, and Zika virus showed that one dose could actually, protect rhesus macaques against Zika. And then component 4 was the HVTN. We started in '99 in a small organization. By 2020, we had 72 clinical trial sites. We had Peter Gilbert, Julie McElrath was our laboratory program, also had David Nominee-Fiore, which we had validated the pseudovirus assay, and we just modified that for the CoVPN, and that's been the Operation Warp Speed lab for defining neutralization. And we had all these really quite prominent international investigators, Linda Gale Becker, Jorge Sanchez, and Beatrice Grinstein. So, great, but I'll give you some personal reflections. Not everything in life is peaches and cream. How did we get all the pharma companies to cooperate? We had HHS, we had BARDA, we had the NIH, we had the FDA, we had the Pentagon, and then we had this guy. Now, one of the things that we did need was, and was really important in this, at least in the vaccine field, was what I'll call the discussion group. Francis Collins came up with this ACTIV, the Accelerating COVID Therapeutic Intervention and Vaccines, called ACTIV, which he came up with in April, and as a sort of a discussion group in which you took public-private partnership to coordinate the research strategy and prioritizations. I got to work with Doug Lowy in the ACTIV Vaccine Working Group, which was a constellation of the company CSOs, the company CMOs, and then had academia, NIH, BARDA, and FDA staff. And we met weekly, starting from the week after he formed this until August, to essentially discuss every aspect of issue of COVID-19 vaccine approaches and trials. This is how we harmonized and socialized ideas. We wrote some major position papers on human challenge, enhanced disease, harmonized endpoints, uniform monitoring, everything. And it allowed everybody to interact, allowed the FDA to hear what the companies were hearing, the companies who heard what the academics say, and it is essentially how we were able to freely exchange information and allow companies to talk to each other. Certainly the companies were not angels. When Pfizer came, I actually talked to them the first, and they said, we want nothing to do with you. You guys are too slow. Now, they started one day ahead of us, actually, but that's it. Their original plan was to do an 8,000-person study. Now, they heard Peter and I present to the ACTIV group, both in April and May, that we were doing a 30,000-person trial. So we then find out that in June, they announced a 28,000-person trial. They added the country of Colombia, and they expanded the trial of 43,000. And then were wanting to get involved with us. And they asked us to use the clinical trial sites and asked us access to the registry, and they became on board. And frankly, they've learned and they've executed. They have been the leaders. We just, are getting pediatric vaccination, as you heard yesterday, because of Pfizer, and they've done a beautiful job. But they did listen. I would get dashboards. Because of time, we would get these dashboards every day of the trial's progress. It's amazing to be able to see a trial in which you're enrolling 1,500 people a day. And to see, and then you get the demography, et cetera. It was really one of the great experiences of life to sort of see these dashboards and see this happen. But the CRO sites enrolled 80% white Caucasians, and they were just pouring them in. And of course, they had they're getting paid on a participant basis, so it's ka-ching, ka-ching, ka-ching. And we in the academic community were enrolling 1/3 of their pace, but we had 20% Latinx, we had 12% Black, and so actually the Moderna trial, the last 3 weeks of the trial, we stopped enrollment of Caucasians, and we enrolled it, so we had the demography of the country. And here's the result from the New England Journal, that that there's 177 cases in the placebo group and 10 in the Hispanic community. And there was 41 versus 4. So we were able to actually show among these communities that the vaccine worked. And that is a very important concept. All right, a little bit more. Politics, science, and election and vaccine. Probably the hardest part of this journey for me was the time of September and October. The trials had started. I had no control over it anymore. I was just looking at the dashboards. It's just under the DSMB control. Pfizer's DSMB was Pfizer's DSMB. Ours was the NIAID DSMB. And the issue was, would there be an early read? And to me, the integrity of all medical investigations that we use was at risk here. This trial and everything we did was in the paper every day. And so the FDA had already been politicized. Steve Hahn had approved hydroxychloroquine on what is absolutely no data. And for me, there was all sorts of stuff. The Biden administration, the Biden people were calling, when are the trials going to read out, this and that. There was really, for me, an interesting issue is, my role had to be as a physician and an investigator to make sure that we preserve the integrity of this, but yet people were dying, so we needed a vaccine. So how do you balance that, and how do you balance that, your own role as a citizen? So, here's reality, okay? Presidential election on November 3rd. The first interim efficacy data from Pfizer was November 9th. The first interim from Moderna was the 16th. Now, I'm going to sort of say, gee, is that just chance? So the politics of vaccine development was keeping the scientific integrity of this grand endeavor, which is a code word is how do we make sure the election doesn't abort the process and initiate an early release of the data? The characters here are the White House, Tony, Debbie Birx, the FDA, the DSMB, the academic investigative community, the company CEOs, and the public. And my comment is sometimes life, as I said, showed you then that slide, has a way of working out. Now, we put into motion two strategies. One was to have a very public symposium in which we would get both the regulators and the press about the importance of the scientific enterprise. First strategy was to go into all the medical schools, or actually all the universities, and see if they would sponsor that. That wouldn't work. There were too many governors of red states who are now presidents of universities. So we actually were very lucky, and Anamari at our university, and I worked with a fellow by Chris Beyer at the Johns Hopkins, who had the website, the Johns Hopkins website, which everybody clicked on, which gave us a forum to hold this. And so we held this symposium, sponsored by the UW and Johns Hopkins in early October. And I'll talk about that in a minute, but it was a stellar symposium, but the target of that was the FDA, Peter Marks, to flush out the issue of a public statement of how long would there need to be a safety follow-up in the cohort before you could action any efficacy data. So what would the FDA require as a safety duration for emergency use authorization? There were no prior guidelines as we were doing this the first time. If you have a BLA or a product licensure, you need 6 months. How long would it be for an AUA? And the second is could we get the companies themselves, especially the lead companies, Moderna and Pfizer, to buy into the idea that early release would not be in their best interest, that no one would use the vaccine and it would damage their reputation and their reputation for all the other things they did as to the veracity of the entire scientific enterprise. We used number 1 to smoke out number 3, and by gosh, number 2 happened. And the drug companies pledged to stand with science on coronavirus vaccines. Moderna and Pfizer actually put out the protocols and allowed the public to sort of see, when the interim analyses were seen. And Mr. Trump didn't like that. The symposium was in early October. And, Peter, we had Francis and Tony and Monsef and all the other people, and Scott Gottlieb, lots of prominent people here, but it was Peter who we were after. This was one of my slides, that the elephant in the room was the EUA from an early interim look, the good from the detection of partial efficacy with the importance of getting definitive knowledge. And about 2 days before that, he made a public statement at the symposium that they would require a medium of 2 months follow-up of 10 to 15,000 vaccinated people for evaluation of any vaccine. This is the dashboard on that day. And if you look at that, we had sort of 20,000 at that point in time. And you go 60 days from them, you really see it's around Thanksgiving. So, post that, we start seeing that the Moderna CEO and the Pfizer CEO wouldn't seek vaccine authorization before mid-November. All right, we're in December. We're feeling good. The vaccine has huge efficacy. The virus, it's an RNA virus, doesn't look like it's changing much. But it then has a gambit. Ancestral becomes now alpha, beta, and delta. Trevor Bedford, who monitors this, is no longer bored. And we have this situation. Black is ancestral, blue is alpha, and everything in life now is orange is delta. And delta is really different. It's R0 is in unvaccinated is 5 to 8. It's not 2 to 3. So that's like varicella, almost approaching measles. Its incubation period is shorter. Its infectivity is higher. Alex did a wonderful study in which he showed at the same CT level, if you then take that sample and look at infectious virus, there's 6 times more infectious virus in Delta than there is with Alpha. So the sort of the infectious particle ratio is really very, very different. So transmissibility has been reported even with CT values greater than 25 to 30. So both more infectivity and essentially still infection with very high CT values or very low titers. Nosocomial and institutional outbreaks, even with vaccination rates, were very high. Younger people are now in the ICU because the older people are vaccinated. And breakthrough cases of symptomatic infection are much higher. And we're starting to see waning immunity. This is just some recent data here. This is the IV network that the CDC monitors for nursing homes. And you can actually see 74% in the pre-Delta area is now at around 50%, the same thing with Moderna. I think this is very strong data because this is the Moderna trial itself. So the Moderna trial, this is the group that got vaccination early. The placebo group were crossed over in January. So the entire cohort was vaccinated, but there's a group that were vaccinated a year ago, 13 months ago, and a group that was vaccinated maybe 6 weeks ago. This is the total number of COVID cases on the trial. So you sort of see that it's perking around at 10 to 15 a month till July. Then you get 81 cases breakthrough And this is the entire cohort here. So, and then 169 cases. So when you look at that as to those who are earlier vaccinated and not, and this is the COVID cases per 1,000 patient years, you see that it's significantly better, or higher, not better, worse, in the early vaccinated group, 13 months versus later vaccinated group. So there's a drop of efficacy essentially of 40% in this cohort. Sort of like, it's, this is a randomized RCT, so the people are really, matched by demography and race and everything about it. And so, you don't know what their daily behavior is now, post-vaccination versus not, but it's as close a match as you're ever going to get behaviorally. So you really do see that there's a marked, and it's probably behavioral disinhibition, because these are the old guys, and we don't take risk. So there's only about an 18% loss. In that. So, boosters, pro-boosters. You can read this, why pro-boosters? It's right here. Here are you guys, if you got Pfizer, which a lot of people at the U did, and all of us at the Hutch did. So here's post-dose 2. You would be at 8 months post 2. This is the ancestral strain. You'd have a titer of 80 if you're you younger people, and it'll go up to 2,000 with the Pfizer boost. Me, I'm over here. I had a titer of 40, but I luckily went up to 2,000. So I'll take 2,000 over 40. How about Delta? This is a different cohort. So this is a sooner cohort, so this is higher than what it normally is. But you divide it by 2 for Delta. It's about 2-fold difference, so it goes up to the 1,500. Moderna is the same way. You do have better durability. So here it is at 198, and it goes up to 4,500. It does make a difference. Recent data about this, if you do get boosted, this is short-term data, but this is from Israel. This is a cohort of 1 million people that they've boosted, a lot of person years, but a lot of cases and a lot of severe cases. They define severity a little bit different. It's sort of like going to the ER is severe, but there's still a lot of difference. This is a 19-fold difference, and you can look at the days post-boost and you see this. Now this is short-term. We'll have to wait and see what happens here. Mixing and matching what your boost should be. This is actually hurt the AD26 program, but here's AD26. This is J&J boosted by Moderna. Here's J&J boosted by Pfizer. Here's J&J boosted by itself. There's clinical data that you boost the efficacy from 70% to 90%, but it's not an antibody-mediated vaccine. It has a better CD8 response and a better CD4 response and a different durability. But at the moment, of the 15 million people getting Janssen, so far there have been 10 million doses of boosting of RNA vaccines. There's only been, I think, 75,000 doses of J&J being boosted. So once the press got a hold of the mix and match, even though it's 50 people, and there's a lot of clinical data on 2 doses of Janssen, it's taking a beating, shall we say. My own impression is that boosting is not really a boost. I mean, most vaccines we give, 016, 012, we gave it really compressed because of an epidemic. We're delighted with the early response. It really helped. But to me, the 3rd shot is just part of the regimen. I'm hoping, and we'll see whether it works, that it's not just for symptomatic disease, but if you look at seroconversion or protecting of acquisition, which would be a really important population effect, we see about 40% of that with the RNA vaccines when it's at the 400 level, which is like, the 2-dose priming. I'm hoping when we get up to 2,000 that we will see this be at the 60-70% range, which would really have a population effect of decreasing acquisition. I don't think it's out of the realm of possibility. To me, it's the main reason why we pushed for the boost and why we want to boost nearly everybody as well as kids, that you'll have more of a transmission effect. All right, lessons learned from all this experience is that it's about people. For the 72 people on this picture who worked in the CoVPN, I just only have admiration for them. And yes, stress, and yes, we have had some fallout from working at this kind of pace during the period of time. I do think there are lessons learned from this success story. When coalitions are made and everybody has skin in the game and a common purpose, you can really move mountains. And you can do it with veracity, with speed, with accuracy, and effectiveness. A key ingredient to success of the country is involving the citizens of the country. And that's something that we need to learn and research, is that the public is involved. You can actually move mountains. And you can move mountains when you work differently and you don't leave things sitting on your desk. And they'll sort of say, well, let's have a meeting in a couple weeks to think this over, or in the next month. And we need to really rethink about things that you do that are important in the care of people to not operate this way. Marketing does work. And we don't do enough of it. We don't devote enough of it because it is actually, if we want to translate medicine and you want people to enroll in trials, 3 to 4% of cancer patients enroll in clinical trials. That's why cancer is so broken from a translational point of view. I'll say that from my view of it. And yes, funding is important. We had unlimited dollars here, okay? But a lot of it went into the manufacturing, et cetera. I saw a slide the other day from Gary Disbrow, who's the head of BARDA, and he said it was $47 billion, 33 went to vaccines. And I'd say each of the trials probably were $1 billion and about $1.2 billion. So we did 5 of those. But the manufacturing got $25 billion and we got 6 or 7 billion. That's still a big number, billion. But that's what was required to do this. Research can be scaled and the involvement of minority communities can be achieved. There's no reason you can't do that nowadays. So cadence of research is important, if not critical, for success. And we as investigators need to involve the public. But cadence is important. If you're going to do a trial and it's going to take 2 years to get the answer, no one's going to give a damn about the result other than the paper. And so, and that can be good in some fields. I got a lot of papers. And that's the cadence of our academic community. But that's not the cadence of improving people's health. And how do you balance that? So we developed great tools, vaccines and monoclonals, yet we're still having enormous death rate. And we also have another only in America issue with us. And that is when we look at COVID deaths and whether it's red and blue, we see that the COVID deaths are red. We have, for the first time in science, an inability to believe or deliver a vaccine that is life-threatening based on your political beliefs. That is not good. This is, again, from the Trump issue. And so how do we tackle the politics in health? I actually don't have an answer to that. And I leave that as a question. I'll only thank the people I worked with. Mike Combe from UNC, Kathy Newsel, especially Mike, who helped me so much early. John Mascola, who is like my brother and continues to be, if I don't call him every other day or every day, it's sort of like, gee, what am I, where are you? David Montefiore in the lab, and I got to work with really great people. David Kessler now, who's the head of the CSO of CAG, and of course Tony I didn't put here because he's on television every day, and everybody knows his involvement. The HVTN executive management team also, who really has worked with me, Glenda, Dan Baruch, Julie, of course, and the statisticians, Peter, Holly Janes, and Yunda, Peter and Holly, who really did all the work in designing these things. And of course, Beth and Mindy, who made all these slides. Thank you. Speechless and breathless here, okay? Yeah.
I actually had a class talking about the COVID vaccine on Tuesday, and we brought up that we might have to end up making the COVID vaccine yearly, like we have to do with the flu vaccine. What are your thoughts on that?
My thoughts are I don't know. The decay curve in antibody after the second dose versus the decay curve after the third dose. The third dose will put more antibody-producing cells in your bone marrow, and certainly going to, produce more memory cells. It may not change the breadth of those memory cells. So the answer is there, I don't know from just a virological point of view, or not an immunological point of view, and I also don't know what the virus is going to do. No one would have ever predict what happened. Could it possibly get another mutation that can make it even more infectious than Delta? I don't even think that what we think is the cause of Delta. It sure is more transmissible, but why is that is really unknown. And we're dealt with this issue that the virus is sweeping through, and by the time you order the virus and get it stocked up and you want to do some experiments, we're onto another strain. I mean, just how I look at how worried I was about Beta because of how neutralization resists, but Delta overtook it. Now, will we get, Beta back or Delta with something? There is this new mutant that we're watching in the UK. We'll see what happens with that. So it's very hard to know. I do believe that we have the surveillance system set up. I do believe we have the tools. We can make a variant. We can boost with that variant, but I don't think one's doomed to yearly vaccination. Now, I think we are doomed to have endemic COVID, and I don't think I think we're going to have to learn to live with this, and I think once protected, we will, quote, learn to live with this, and I hope a year from now, this auditorium will actually have 200 people in it rather than 30, and, like the old days. So I do think we will get there, and we'll get there because of science. It is the biomedicine and science that's going to do this.
And a few questions online. One from Lily B. Is the structure used to recruit BIPOC people in these vaccine trials going to be used in other clinical trials?
Well, I can't predict. I would say in the HIV business, we've been able to do this in the last 8 or 9 years, and I think it's a lesson learned. At the very top, Francis and Tony and everybody recognizes that we had done this. We have been working with other NIH programs. I do think that pharmaceutical industry has looked at this and held their feet to the fire, and they have responded, and that the CROs started doing this. So I'm hoping that this will, translate. But it does mean building trust and doing that, and that is not something that just happens, okay? It requires work.
Was this infrastructure leveraged to make monoclonal antibodies, or was that a separate?
The infrastructure was leveraged. I mean, we had the AMP trial that actually, showed that a monoclonal could prevent infection. And the original humanized infectious disease monoclonal came from the VRC. So, and then Ebola came from the VRC. And so, I think the whole monoclonal business, as it relates to BARDA, and when that became sort of the best biological approach to actually, if someone would, to give antibodies, especially when vaccines, for non-vaccine preventable diseases. So that was leveraged. And the whole monoclonal business in the pharma industry to be able to manufacture monoclonals at scale. Can look at it, still say it's not what it should or could be, but, millions of doses have been given. And so the answer is yes.
Yeah.
So, Larry, you made a great case about just the unprecedented nature of this response. So fast, from knowing nothing to a vaccine in less than a year. So we have this vaccine platform. We've got these great clinical trials. We've got tremendous diagnostic capabilities, sequencing. Is there an unexpected byproduct benefit from this? In other words, are other areas of medicine, of infectious disease going to benefit? And can we find a silver lining in all of this?
Well, well, I hope so. Yesterday or 2 days ago was a symposium, a couple of days, that sort of, lessons learned for COVID for HIV. And, and what, got me to thinking about, how do we change that? And I do think RNA technology, for a lot of traditional infectious disease or things that we don't have vaccines for, suggests that RNA technology should be used. And, You can make RNA now make a ferritin particle. You can make with a protein, and so it allows you to iterate things in a way that you've not been able to iterate before. I also see that we'll start distributing all this stuff about how do we make RNA for the world. This brought up the politics of nationalism, vaccine nationalism, which you can look at and sort of say, I'm not sure that's ever going to be overcome. But how do you overcome it? Well, you get manufacturing on every continent, in many places on every continent. So, so they can make the vaccines that are, important to them. Because the cost of goods is not expensive, okay? It's, $5 to $6 to do this. And, it's not that expensive, and okay, it's all synthetic. You don't need big vats. You can make in one place, you can make, a lot of RNA, bottling it and all that other stuff. But regionalization is needed, and I do think it's going to happen in the next 4 or 5 years. Generic regionalization of RNA technology. Yeah.
So Susan Fink asks, can you please comment briefly on the correlates of protection and the role of neutralizing antibodies, T cells?
Oh, I didn't have time to do that. I cut those slides. There is a preprint from Peter and MedRx that does the correlate of protection for the Moderna trial. Neutralizing antibodies, even if you don't have them, it explains about 50% of the correlate of protection. So you get a 50% effectiveness if you have no antibody, but very few people don't have antibody. It's about 70% if your antibody is in the teens, like in the 40s, like I said. You get to above 100, it's 90, and if you get above 1,000, it's 97%. So that's sort of what it looks like for RNA. But, that sort of suggests low levels of neutralization, which we're not consistently detecting could be partially effective. That's all with the ancestral strain. I don't think that curve is accurate for Delta, because we're seeing, I showed you the data there. So I think that one of the issues with correlates of protection is that we have strain variation. So extrapolating what happened with ancestral strain to Delta is not, I don't think is correct. Okay, I think you're going to have to go up to where 70% is probably at 100 or 60%, and it's going to, you're going to be in the 2000s to get into the 90s. Don't quote me. I mean, don't say Larry said, oh, that's data. That's opinion, okay? That's not a curve.
So we're running out of time, so one more question from online. Is it better to get a different brand vaccine for the third shot or not? So in a
I got boosted with Pfizer. I started out with Pfizer. How was it? And so I had no qualms about that. I'm sitting there on data saying, yeah, Moderna is probably a little bit more potent. You get 100 micrograms, it's probably better than 30 micrograms. But, I had handled the prior doses just fine. Everybody will make the government has allowed it to happen because we wanted actually convenience. So, so you wouldn't waste all the vials and, so if you want Moderna, you can get Moderna, or if you show up at Rite Aid and they have Moderna and you show up at Walgreens, they have Pfizer, get it in your arm. It's not going to make that much a difference. The strand, the 2SP strand is identical.
Get it in your arm sounds like a good bite 10.
Okay, get it in your arm. All right, thank you.